Hello students, we were seeing the chromosomal theory of inheritance. In the last class, I told you that the chromosomal theory of inheritance is also called Boveri-Sutton chromosomal theory, and it states that the pairing and separation of a pair of chromosomes would lead to the segregation of the genes. So earlier, Mendel told that the genes are segregating, but according to this theory, they say that the chromosomes are segregating. Because the chromosomes are segregating, so the genes are also segregating. So that is what was said by Boveri-Sutton chromosomal theory. So this was a theoretical definition that was given. So in order to confirm it, there was an experiment conducted by Thomas Morgan. He conducted an experiment using fruit fly. The scientific name of fruit fly is Drosophila melanogaster. So Thomas Morgan, he conducted an experiment using fruit fly, and that is why he is called the flyman of genetics, because he conducted the experiment using fruit fly. That is why it is called the flyman of genetics. This fruit fly, why is it called fruit fly? Because it is mostly seen to sit in banana and mango trees, fruits. And what is there inside the fruit? There is auxin. We have studied auxin. So auxin helps to produce yeast, and because of which this fly is attracted towards the yeast to eat the yeast. So that is why it sits on those fruits. So that's why it's called fruit fly. Now, why did Drosophila choose this? Why did Thomas Morgan choose Drosophila for his studies? Because the first reason was that it could be grown in laboratory. It could be grown in laboratory on simple synthetic medium. The culture that can be used was simple for growing it. They complete their life cycle in 2 weeks. They can complete their life cycle in 2 weeks, and it was also Thomas Morgan later also said, when he was experimenting, he also said that it can be cultured for two more weeks, like 4 weeks. It can be kept for, so that it can be studied. Its life cycle can be studied. Then a single mating could produce a large number of progeny. Like a single mating on fertilization can produce large number of progeny flies. So large number of progeny was produced in a single mating. Then the male and the female, they are easily distinguishable. Okay, that's another feature why he selected Drosophila. Then it has many types of hereditary variations that can be seen with low power microscope. The hereditary variations can be easily observed with a low power microscope, which made its study easy. And it had 4 pair of chromosomes. 4 pair of chromosomes, 3 autosomes and 1 sex chromosome. So it had 4 pair of chromosomes. These were the reasons why he chose Drosophila for his study. Now, what did he do? Morgan bred many generations of fruit fly, and Drosophila normally they are red-eyed. Normally Drosophila is red-eyed male and brown-eyed red-eyed male and brown-bodied male. That means the male would be red-eyed and brown body. Okay, and females are white-eyed and yellow body. This is the general. Generally, Drosophila would be, the male would be red-eyed and brown bodied, whereas the female is white-eyed and yellow bodied. How you can remember? You can remember that like girls are usually white. Okay, so you can consider a white girl in a yellow frock. So white-eyed and yellow, that is yellow-bodied is female. Here you can have the brown boy, brown. So brown body, red-eyed. So a brown boy with a red shirt. Okay, you can remember that way. So normally Drosophila is red-eyed, brown-bodied, and white females are white-eyed, yellow-bodied. So when he was culturing the fruit fly, he saw a single white-eyed male fruit fly. So he observed a single white-eyed is usually female, but he observed a single white-eyed male. So what did he do? He crossed this white-eyed male with red-eyed female. He got red-eyed female also. He got white-eyed in the culture. So what did he do? He crossed white-eyed male with red-eyed female. So when he crossed that red-eyed and white-eyed female, when he crossed that, suppose we take red-eyed as XW plus. XW plus. This is for red because it is dominant. Okay, and white-eyed we take as XW, and since it is a male, XY. XW plus for red and XW for white. So when we cross it, when we see its cross the F1 generation, we see we are taking red-eyed female. So XW plus, and we are taking male XW and Y. Okay, so XW plus and XW, that is one XW, then XW plus and Y, XW plus and Y, XW plus and XW, XW plus and Y. So XW plus XW plus, that is the dominant character. XW plus and XW, that would become red-eyed female. XW plus XW again becomes red-eyed female. So these are red-eyed female. This also red-eyed female. XW plus and Y and XW plus Y, it becomes red-eyed male. This becomes red-eyed male. Okay, this is for the F1 generation. Now we will see in F2 generation what happened. In F2 generation, we got here red-eyed female and red-eyed males. Okay, now in the F2 generation, again red-eyed cross between in the F2 generation, the cross between red-eyed female that we obtained in F1 generation crossed with red-eyed male. Okay, so what happened there? Now red-eyed female, how we can write it as XW plus XW. This is what you got in F1 generation. And red-eyed male, XW plus Y. Okay, now when we draw the Punnett square for this, XW plus XW and XW plus Y. So here XW plus XW plus XW plus Y, XW plus XW, XW, Y. So this becomes red-eyed female. This becomes red-eyed male. This becomes red-eyed female, and this becomes white-eyed male. So this character, that is, here you can see it was red-eyed female and red-eyed male, but still white-eyed male came here. Okay, so here it was observed that all females, this is female, all females are red-eyed. Females are red-eyed, whereas male can see 50% red-eyed and 50% are white-eyed. So what was the conclusion that he draw? He came to a conclusion that the mother passes the trait to the son and the father passes the trait to the daughter. Okay, so here the mother is passing the red-eyed character, the mother is passing the trait to the male, the red-eyed character, that it is passing the trait to the son. So this showed that the genes located on X chromosome, so the genes are located on X chromosome, for which there is no corresponding allele on Y chromosome. That means here all the changes that are taking place because the genes are located on the X chromosome. Okay, so this is what is called the sex linked inheritance. Sex linked inheritance. What is that? The genes located on X chromosome, okay, for which there is no corresponding allele on Y chromosome. There is no corresponding allele in the Y chromosome. So only the X chromosome is the one which decides the character of the progeny. So this is called the sex linked inheritance. Okay, so the transmission of gene take place from one generation to another through the genes that are located only on the X chromosome. Y chromosome is not playing any role as such there. So this is called sex linked inheritance. Now coming to the next topic, that is linkage. We will come to the next topic, that is linkage.